research of this multidisciplinary webinar. There is a one saying in Indian tradition, Ano Badraha Krutah Yantu Vishwataha. That means all the good and progressive thoughts are welcome from entire part of the world. So I welcome you, sir, on behalf of Sakarchand Patel University. Namaste.
It's a pleasure.
Thank you, sir. Now I would like to welcome Dr. J.K. Patel, sir, Dean, Faculty of Pharmacy, to give a welcome address.
Thank you, Dr. Hirak. Good afternoon, all, and good morning to Professor Enzo. Chief Patron, Honorable President of Sakarchand Patel University and Chairman of NSVKM, Sri Prakash Bhai Patel, sir. Patron, Honorable Provost of the Sakarchand Patel University, Dr. J.R. Patel, sir. Honorable Dr. D.J. Shah, sir, Technical Director, Technical Courses, Sakarchand Patel University. Today's keynote speaker, Professor Enzo, from the Department of Life Science and Environmental Science from the Italy. Dean External Affairs, Dr. Anil Managutti of the Sakarchand Patel University. Organizing Secretary and Coordinator, Dr. Neeraj Joshi. Faculty members, participants of the online e-webinar of Sakarchand Patel University in multidisciplinary international series, and my dear student. By virtue of my being a head of faculty, I am here to bid you a pleasant welcome and brief you about the institute. Our institute is dedicated to imparting the quality of pharmaceutical education and promotion of the research qualities among the student as well as the faculties. Institute has completed about 12 government as well as industrial sponsored research projects sanctioned by various government agencies of the Government of India and Government of Gujarat. Till date, Institute has received more than 2.5 crore research grant from the various government agencies, and more are in pipeline and considerations. Topic is itself interesting, and it is hoped that in the depth discussion of the topic by resource person like Professor Enzo will positively benefit us all to very great degree. Once again, I bid pleasant welcome. Thank you.
Thank you.
Thank you, sir, for your kind and motivational words. Now I would like to request Dr. D.J. Shah, sir, for a keynote address.
Good afternoon to all, and good morning to Dr. Enzo. Really a pleasure to be a part of another multidisciplinary webinar that is organized by team of Dean External Affairs, Dr. Anil Managutti. I'm really thankful to our Honorable Provost, Dr. J.R. Patel, and our President, Sakarchand Patel University, Sri Prakash Bhai Patel, for helping us to start this kind of series, which helps actually researchers all across the world to interact with each other and then share the knowledge. I'm going through the file of Dr. Enzo, and it's really interesting that you've been working in the area of virology and life sciences for a long. So I'm sure that this interaction that the discussion that you are going to share today is going to help all our students in understanding different methodologies used in study of virus across like different domains. And this interaction, along with the different approaches that you follow, can help society to fight with availability of drugs to fight such kind of diseases in future also. So I'm sure that this is going to be a useful interaction, and even AI-based algorithms on understanding the cultural behavior of the various genome sequences also helps offline studies on a computer or simulation-based study to derive different results, because then you are not practicing with the human being or the live organisms. So that also helps. So Sakarchand Patel University is always ready to carry out this kind of research along with the pharmaceutical research that is actually happening. And this interaction will definitely help all the students as well as the faculty at Sakarchand Patel University to understand it better way. So thank you very much, Dr. Enzo, to be part of this seminar, and thank you, Team SPU, for arranging this seminar for the benefit of our students and faculty at the university. Thank you very much.
Thank you, sir, for motivating us. Now I would like to request Honorable Provost of Sakarchand Patel University, Dr. J.R. Patel, sir, to say a few words about this webinar.
Morning, Professor Angel, and good afternoon to all participants in today's international webinar. It's organized by a team of external affairs and faculty of pharmacy. I must say that our university, as Dr. Vijay Shah said, Sarkar Patel University is very ahead for having this kind of program for the benefit of our student or faculty and other people. As far as research and all aspects concerned, for that purpose only, and as well as for the faculty exchange program, we have done MOU with the University of Italy, and this is the first kind of program which is organized by our team. Under that MOU which we have done, and I hope in future many more programs also will be there for the benefit of our student and our faculty. We will have a student and faculty exchange program also, so that we can strengthen the research aspect of our university. And like Vijay was telling that for the benefit of our student institute, also that will lead or other will add into the growth of our university as well as our work constituent institute. So once again, I congratulate the whole team for this kind of program, and I am thankful to Professor Anjo for accepting our invitation for delivering the talk on this pharmacy related the program whatever we are doing today from his deliberation. And I am thankful to our President, Sarkar University, Sri Prakash Patel, who has given boosting to all our faculty, all our officials for conducting this kind of program, and for MOU also. We are thankful. All officials from the University of Cagliari, and definitely I assure Professor Angel that in future we will be having many more programs under this MOU. So all the best for successful program. Thank you very much.
Thank you, sir. Now it would be my pleasure to introduce respected Dr. Enzo Tramontano, sir, on this international webinar. Sir is working as a professor of microbiology at the Department of Life and Environmental Science, University of Cagliari, Italy. Sir is also professor associated to the Institute of Genetic and Biomedical Research of National Research Council in Cagliari. Sir is ambassador for Italy of International Society of Antiviral Research. Sir is a member of Executive Council of Italian Society of Virology, American Society of Microbiology, founder, member of European Society of Virology, Italian Society of Virology, New York Academy of Science, Italian Society of General Microbiology and Microbial Biotechnologies. Sir has published 5 patents and delivered more than 30 lectures in various international webinars, seminars, and development program. Sir is also an editorial member of Antiretroviral Drugs Development and HIV Cure Research Microbiology Section Virology in ACTA Microbiological Burguria Journal. Sir has published more than 121 research publications in various reputed national and international journals. So it's our pleasure to have a great scientist like Dr. Enzo with us. So I request sir to take a lecture on this multidisciplinary webinars on DNA replication of drugs on SARS-CoV-2 COVID virus. Thank you, sir.
Thank you very much for your kind invitation and for this very kind introduction. And so I'm very happy, I'm very glad to be able to give you a lecture of what we are doing now and to, to explain the students what we are doing and also to show all of you our work in just in view of possible cooperation that are always very welcome and for both for research activity, but also for teaching activities. Why not? So we are involved in a number of cooperation, also of not only, of course, of research, but in the last years also on teaching with the other university, especially in Asia. We work a lot with Taiwan, but we are open to any kind of university agreements. So maybe we can discuss it later. So I would like to start my presentation. So I try to share my there we go. I should share my there we go. I think you should see it, right? Okay. So yeah, perfect. So the topic of my lecture today will be approaches to develop drugs to inhibit SARS-CoV-2 replication. My laboratory has been historically involved in their development, mainly, let's say, on HIV. I mean, we have worked many years on HIV development, and then we shifted to other viruses. In particular, we shifted to find drugs against Ebola virus. And at the same time, we opened a research line on human endogenous retrovirus that have implied a number of cancers. And then, as all the world did, basically, we got stuck in this pandemic. And so, of course, in the last 2 years, let's say 90% of my laboratory activity has been moved to study and to fight SARS-CoV-2 replication. So my lecture scheme will be the following. I will give you a very brief introduction of SARS-CoV-2, just to remind students, they probably know, I mean, a few basic molecular virology items. And then we go, why do we need antivirals? What is drug repurposing? What is the two main approaches used today? And then a final remarks. So let's start with introduction. As you know, coronavirus is, the SARS-CoV-2 belong to the Coronaviridae, and it's basically, it's an nidovirales order, so Coronaviridae, and particularly Coronavirinae, that have 5 genus alpha, beta, sorry, 4 genus alpha, beta, gamma, and delta coronavirus. Okay, as you can see here, you have alpha, you have beta, you have delta, and you have gamma. And so there is a big diversity among them, even though, of course, they belong to the same subfamily. So it's, of course, they have a big similarity, but there are differences, important differences. And as you can see here, this is the phylogenetic tree of many of them, if not all. And when there is this black dot, you can see there are the human coronaviruses. So just to say that human coronaviruses are, you have both in alpha, beta classes, and the red dot is, of course, the SARS-CoV-2.
Hello, sir.
Yep.
Hello.
Yes. Is it okay? Do you hear me? Okay.
Yes, sir. Yes, sir.
Please continue, sir. Continue, sir.
Okay. Okay. So sorry. And so, and as you probably well know, coronaviruses are well known in veterinary medicine. So normally they are diffused in animals. Okay, wild animals like birds, like bats, like rodents, like pangolins, civet, but also in farmed animals like swines, like cattle, horses, let's say, and also like pet animals like dogs. Okay, from the a number of animals, okay, you can have the passage of the virus from one animal to the other, and then at the end to human. And this has been happened a number of times for what is called the alpha coronaviruses, like NL63, 229A, E, sorry, okay, OC43, and then of course for SARS-CoV-2.
Hello, sir. Your voice is low.
I try to speak louder, but I cannot speak louder than this. Sorry, I'm not sure how to handle this, to be honest. And so the human coronaviruses, that have been 7. 4 of them, the 229E, OC43, NL63, HKU1, are endemic in humans and are mainly associated with mild respiratory illness. Okay, so they are been around for many decades, as far as we know, probably more. Okay, and they can determine severe acute respiratory. And then there is the 3 more let's say severe illness, like the SARS-CoV, as you know, the MERS, and the SARS-CoV-2. Okay, these basically, as you see here, 4 of them, they give mild respiratory tract infections, 1 gives pneumonia, and then, you know, the 3 they are more dangerous. Have different, I think you know all, SARS-CoV in 2003 had a big, it was discovered, but there's a 10% fatality rate. The MERS-CoV that started in 2012, that they still have, is around, that has 37 fatality rate. And then the SARS-CoV-2, which of course, as you will know, it's not clear where they come from, but you know, has a fatality rate probably around 2-3%. So what is the genome of the SARS-CoV-2? The genome is as RNA genome, positive strand. So it has a cap in 5' and a poly A and 3', and is a long RNA, because it's like 30,000 nucleotide RNA. So just remind you that HIV has a 10,000 nucleotides as a length. So here we have 30,000 nucleotides, so it's much longer. And in fact, it codes for many proteins. The proteins are basically coded in different ways. There are a number of proteins coded directly when the virus enters by ORF1A and ORF1B. So ORF1A includes basically 2 proteins, PL-PRO and 3CL-PRO, and a number of other accessory proteins. Then there is a ribosome shift here to allow the production of ORF1A, 1AB, and that includes other proteins like the RNA-dependent polymerase, the helicase, and a number of methyltransferases that are important then for the replication of this genome. In fact, once this RNA has been translated by the ribosomes, the translational system, the complex start to replicate the minus sense, and from the minus sense, there have been a number of let's say linear translation, so that you have a number of RNA that are short, from which they are made of plus RNA that goes to synthesize other protein like the spike protein, the N protein, the matrix protein, E protein. So a number of proteins that are, let's say, important for the viral construction. So this is called discontinuous transcription. So, and on the other side, there is also a continuous transcription that give you a minus RNA, genomic RNA, from which then there are many copies of plus RNA. That is the genomic RNA. As you can see here, there are a number of proteins because, as I was mentioning you, the virus is a big virus, has to be an RNA virus, and among the very important thing that is among the proteins that have been synthesized, produced, there is also a proofreading activity that is very important, because when the viruses get so big, RNA virus gets so big, the viruses need to have a control, a proofreading activity. This is a general picture showing you not only the viruses, but also the cell cycle, the virus replication cycle. So you have the entry, as you know, the entry goes through the interaction by the spike protein, between the spike protein and the ACE2 receptor, and most probably it enters like an endocytic mechanism, even though it's not impossible to envision also a fusion of the level of protein. And then, as I was mentioning, you have as the first thing the translation of the 5' protein, 5' portion of the RNA genome. So you make a number of proteins that you have a polyprotein that is cut by viral proteases. So you generate, there is a generation of the replication complex that start to synthesize genomic and subgenomic RNAs. All these, even though you see it here, really it has been shown recently. This process takes place within some double membrane vesicles, from which then the RNA gets out. Okay, so you have more production of more proteins, and of course replication of more viruses, and then these vesicles mature, acquiring a number of proteins that comes from the endoplasmic reticulum. So you have basically the generation by Hershey of exocytic vesicle that you see here, basically, that are then go out of the cell. So this is the process you see here, the process of the virus getting out, and this is the photo of the viruses. So this is very simply the replication cycle. And if you have a question, I will be happy to answer on you. Here at the beginning of the pandemic, of course, many things were not known, especially because even though the SARS-CoV-2 is very similar to SARS-CoV, however, since that infection was expired, basically nobody really cared about studying this virus. Now it's clear to everybody that we need to enter into the detail of this. And one important thing that I will not very go further, but I think is needed to mention, that of course, you know, all of you know that we have an intrinsic immune activation system that is relevant for the defense of our immune defense, for the first alert. So you have a number of alert system that basically are measured, most of them against viruses, but it's important to note that a number of protein, especially some small protein, non-structural protein, have as an adjuvant further role in addition to their, let's say, role within the replication complex or the maturation of the other proteins, the control of the transcription. In addition to them, there are a number of proteins that go to inhibit the intrinsic immunoactivation. One for all, the protein N. N for instance, N is a protein that is very important for, of course, is a nucleocapsid protein, so it's essential for the making of the capsid, let's say, and so the entry of the genome into the virion. But this protein is also important for shutting down the production of interferon, or, for instance, the protein, the PL-PRO, that is a protease that is very important for the very first cut of the polyprotein. So without it, basically, there is no processing of the polyprotein that is made by the virus. However, this protein is very important also for the processing of a number of genes, cellular proteins that are synthesized to contrast the viral replication, and in this way the virus can replicate much more easily. So just to say that there is really a fight.
Hello, sir.
Yes?
Sir, please remove someone. My friend is in waiting room. He can't join.
Okay, sure. They will let you soon inside. And so these proteins will be cut by the PL-PRO. So this just to tell you that the replication cycle is more complex because there is also interaction between the virus and the immune system. Next, so this was a very, very, very thin introduction to the virus, and I didn't really. I mean, there are many themes that can be deal with, but my talk is on antivirals, so I will try to give more attention to this. First of all, so the question is, why do we need antivirals? We have vaccine, right? So all over the world, vaccine is going to be distributed, and so we have a big surveillance now. There is a tracking system that is being put in place and is decently working. We have a diagnostic system that is important. We have, again, a prevention, that's so vaccination that is working so well. So why do we still bother thinking to antiviral now? Well, first of all, because not all people can be vaccinated, first of all. Secondly, because as you can see, there are many variants that are coming out, and to have the possibility of treating people is also important to stop eventual variants that can be, let's say, escaping the immunity given by number of vaccines, but also because preparing antivirals can save the life of people that anyway got infection, didn't get a good vaccine immunity, or again, they cannot get vaccinated for any reason, that they have a very low immune system, and also because it's a way of preparing ourselves to the next, let's say next possible infection, epidemic, or at least, if hoping not pandemic. So to develop drugs, in other words, if we had thought that the 2012 infection could be replicated 10 years later, well, in that 10 years, we surely have developed some drug that would have stopped the pandemic much earlier and saved millions of lives. So what kind of drug do we need? Well, we need a potent and selective drug that acts both in vitro and in vivo. We need an optimized drug for on-target activity. Of course, we need adequate PK exposure. We need sufficient safety and efficacy doses. We need option for route optimization. We need, of course, a scalable and cost-effective production, and we need something that's suitable for stockpile. This is like our ideal drug. And so, of course, this is what we have to keep in mind when we think to develop a drug.
Hello, sir.
So the question next is, which drug target should we follow? Of course, as I
Hello, sir.
yes. Of course, if you
Manav, please keep your audio mute. So go ahead.
Yes, sir. Yes, sir.
Okay, as I was mentioning you, I mean, there are a possible number of target that can be used, host target or viral target. As a first thing, host target, of course, are probably have a low selectivity, a higher safety risk, and they have a higher though barriers to resistance. A possible host target, for instance, is the cellular receptor. Okay, but of course, the cellular receptor has also a role, a physiological role. So we need to be careful that a drug binding a cellular receptor can, in some way, have a side effect that is worse than the efficacious effect. Other targeted are viral proteins, so they are more target dependent. We have in this case a high selectivity. In general, have a lower risk. We might have a barrier to resistance that is lower than the cellular target, and we have probably very unlikely this is a tissue dependent issue because the virus is always the same. And if we go to see, well, what about the current antivirus against other viruses? Very few are targeted to host proteins, the great majority on the viral proteins. So if we think about the viral proteins that can be targeted, we have the viral entry. Okay, as I was mentioning, viral entry is involving the interaction between the spike and the cellular receptor. So we need to stop this process, or the entry process by endocytosis. And the endocytosis is, I'm not entering into the detail of it, but you need also an acidic endosome. You need also a proteolytic cut of the spike. So, you need another of other issues that can be stopped blocking the entry. Of course, you have, as I mentioned before, the viral proteases. I will mention you later a little bit. So you have viral proteases that are for sure a demonstrated target, validated target. You have a viral RNA polymerase that also is a very good target, explored in many cases. We have other viral proteins involved in replication, other the RNA polymerase. I will briefly mention about the RNA helicase. And of course, you have viral protein involved in what I just gave you an hint of it, the viral protein involved in innate immune evasion that are also a possible target. If these are the target, which are the, let's say, the strategies? Well, we basically have two main strategies. One is called drug repurposing, and the second one is new drug identification. Drug repurposing, of course, means that you have a compound, a drug, basically, already approved, or at least it passed the safety control, but maybe it been discarded during the clinical trials. But drug repurposing basically deals with drugs that have been already passed a control of safety. So either they've been approved or not approved, they having passed the safety control. If you demonstrate that they have an effect on the virus, they can be used immediately, basically. Okay. And in fact, as you can see here, the drug repurposing is much faster than the new drug identification, especially because new drug identification required a long way to arrive to preclinical studies, and you need much longer preclinical studies, first of all, to demonstrate safety and then to demonstrate efficacy. Okay, so basically, the first let's say the first strategy that people have been used until now is drug repurposing. I just want to mention that for now, but as far as today, so we are almost 1.5 years from the pandemic. Only one new drug has been entering, not been approved, but has entered clinical study, and it is a Pfizer drug. Okay, drug repurposing strategies can be a number. Okay, so as I was saying before, they can target viral proteins or host proteins. Okay, and of course you have heard of a number of drugs that can also act directly on the viral. One of the classical repurposing drug that we have been using and we have heard of is remdesivir. That was a drug that has been initially developed for Ebola virus. Okay, so this was an antiviral that is directly acting on the viral replication. But also when we think about drug repurposing strategies, we also think about immunomodulators that either reinforce the immune system, or since there is also, as you know, in case of coronavirus, a big issue about inflammation in the lungs that reduces, let's say, the immune activation in the lungs, reducing so the damages that the virus makes to the lungs. Okay, and of course other drugs that can protect the lungs or elevate the oxygen supply to the lungs. So there are a number of strategies that led to repurposing strategies. I'm focusing basically only on antivirus, and so when we speak about drug identification, either by repurposing or basically repurposing or new drug identification, we have basically two main approaches. What we call a phenotypic approach and what we call a target-based approach is mainly a biochemical approach, while the phenotypic approach is basically a cell culture approach. So the phenotypic approach basically its method, the methodology with which we study the viral replication in cell culture. So we have, of course, BL3 containment, we have incubators, we have flask and multi-wells. So as you can see here, you have an uninfected layers of cells. If you infected them with SARS-CoV-2, we have, after 5 hours, you start to see some cytopathic effect. Then after 8 hours, you see a much bigger phenotypic effect. And in 24 hours, you see really mostly of the cells that are dead. So what you measure is a cytopathic effect, so the killing of the cell by the virus. And we measure it normally in a number of ways, but mainly using some marker, cellular viability marker that are mostly based on the color, let's say, of some, this is an MATT assay. So basically it's very simple assay in which you add a substrate. If the cell is viable, happy, that it will be able to have, to give you a production of, to process the substrate. So you have a nice color, okay? If you have the virus, the cell plus the virus, you don't, the cell is dead. So basically the well is colorless. Okay, so this is the control. This is cell plus cell viruses. The virus is killing the cell. The virus is killing the cell. Okay, when when if you add the drug, you basically have a recovery of the viability. In reality, in our laboratory, we do not use that system. We used some cells, spheroid cells, that are that is normally expressed GFP, that is a fluorescent molecule. So basically, what happens that if you see the cell and then you measure you go to see it, you see a very nice green layer of cells. Okay, so you can measure, basically, you can measure the intensity of the fluorescence that comes out of the cell. Then, if you treat, if you infect, basically, if you infect the cell with a virus, you kill the cell and you have no signal. Okay, so basically, there is a big difference between the uninfected cell and an infected cell. Then, if you treat the infected cells with drugs, then you go to recover the signal because you have an inhibition of the viral replication. Okay, so this is our main system of screening drugs, and to assess the drug against the viral replication. Out of them, we have a number of biological validation using other cell culture, other system, and then we use also other viruses, but mainly we are using now this system to assess in a phenotypic way the efficacy of a drug on viral replication. In SARS-CoV-2 is a European funded project we are currently working in, and we just published a paper. This is called a review, but it's been published, just accepted. And you can see here in Nature Drug Screening, something like this. And so if you see here, we did a drug repurposing. We started from 8,000 compounds. We have 184 compounds that shows a more than 80% inhibition. We confirmed 132 compounds. 70 of them were active with an inhibitor activity lower than macromolar, and 3 had a safety index greater than 5. So overall was not a great success, but was a success. And so then we also did a counter screen because of this, some of these compounds. So this is what we did, and we publish it. Okay. However, I want to mention that it's not so straightforward, this drug repurposing, because some of these compounds were also repurposed, were also studied by other team. We are in the team of SARS-CoV-2. There are also other screens, other consortium, and basically this is the ideal situation which you have a good correlation. In reality, honestly, there is the correlation is much lower, and so basically you have hundreds of active compounds that found active across multiple screening, repurposing screening, but when you go to normalize the library composition, it's very difficult to make an assessment of all these studied together. Okay, so there are differences in them. One of the differences is that the cells respond different to drugs, because some of them, especially the one we use, for instance, have a pump that gets out the compounds of the cell, and so this is a big issue. So we need to be careful. Of course, there is a problem of so the PGP, I mean, we mentioned it. There is a problem of readout. So when do you read? How long the assay are run? Because of course there are different issues. The ideal will be a very short time to assess really the viral replication. Once you have a longer timing, you can have more effect on the cell, but some cells replicate much lower with lower rate, like Cal3. And so there are a number of passages that are different. And so the cellular results can be very different. And we need to compare primary data. And in fact, both European Union and NIH have opened some portals, which we normally upload our results to be able to compare them and make a sense of them, because it's not very straightforward. In fact, after 1 year, no real drug against coronavirus had been found by these Republic screening efforts that was anyway very, very big. The second approach is a target-based approach, in which basically we do not start from cell, because in the phenotypic-based approach, you have basically all the targets. You don't really care what target you are hitting. You just care to stop viral replication, and you measure the stopping by measure the cytopathic effect. And then afterwards, you come back and say, okay, let's start to understand which is the target through which this compound works. In the target-based approach, the approach is completely the reverse. So we start with a very focused target. Okay, so we start from the target. We don't start from the viral replication, but a single viral, normally enzyme. These are the enzymes we are studying in my laboratory. So we have NSP5, that is a protease, TCL, the main protease, NSP2, that is a secondary protein called PL protease. Then NSP8, that is a RNA polymerase RNA dependent. Then we have RNA helicase, and then we have two methyltransferases that are important for the capping, N7 and 2O methyltransferases. I will show you some data on TCL-Pro and some hints on the polymerase and on the helicase. So when we discuss about target-based approach, what do we mean? We mean that we cloned the gene for protein expression, okay? Then we did the bacterial transformation. So we have the gene within a vector. We express it in bacteria. We express the protein, okay? We purify the protein. We verify the purity of the protein. The protein here is pure enough here, and then we can start with some enzyme assay. I'll show you the enzyme assay for the protease. Okay, what we use for the protease is a peptide. This protease, of course, the enzyme who cut the protein. Okay, so we mimic the substrate of in this case, the 3CL protease. So you see, we have a peptide, a short peptide, and the peptide has in one amino acid addons. To this amino acid, we have a fluorochrome that emits some
Enzo.
Yes.
What is it next, sir?
So we have meaning we have a fluorophore who emits light, but close to it. Okay, now you hear me. So someone shut me down. Sorry. So the addons is a fluorochrome who emits light, emits a certain fluorescence, it's a certain wavelength, but normally within the peptide is quenched by a quencher. So when the peptide is entire, basically it cannot emit any light, not emit any signal. But when the protease cut here the peptide in two, then you see that you have a signal, a fluorescent signal, okay? And of course, okay, so once you make this study, then you can have an inhibitor, and you can measure the inhibition of these of whatever compound on this enzyme activity, and you should obtain this nice curve. And of course, this, for instance, is inhibition curve on the SARS-CoV-2 3CL protease. So we can identify some compound on it, but at the same time, we have to ask, well, is this compound inhibiting only SARS-CoV-2 or also other viruses? So we have also the same biochemical assay for other coronaviruses like MERS, for instance, and you can see the same thing. So you have to clone the gene, express the protein, purify the protein, establish an assay, and then study the compound on this system too. What we did, of course, when we do screening system, we have robots to make this faster. So we have output screening systems. So we started, in this case, we published this year on the HES Pharmacological Translational Sciences. We started for around 9,000 compounds. We have around 250 heat profiling. Okay, and then you see these are drug purposes. So we started with different categories of compounds. We came out with a number of compounds that inhibit well the enzyme, the 3CL Pro. Okay, and we also made some crystallographic studies to see how some of them actually inhibit the enzyme, and a number of them inhibit also viral replication. So in this case, you start from an enzyme, okay? You start from an enzyme, okay? Ask whether the enzyme inhibits in this case, the SARS-CoV-2 3CL Pro, the one that are active. You measure also the specificity. So you measure the activity on other coronavirus like MERS. Then you measure the cytotoxic effect in viral cells. Of course, you measure the toxicity in viral cells. And you also measure, since 3CL Pro means chymotrypsin-like protease, you also measure the activity on chymotrypsin to measure whether there is a toxic effect on this protein. And this has been published, so if you want any other information, you can go through it. And this kind of study, we are also doing this kind of screening on the RNA-dependent RNA polymerase, in which we have basically a single-strand template I was mentioning before. So if you put the RNA polymerase complex in the presence of a single-stranded RNA template, you get a double-strand RNA product, and with an intercalator, you can measure the production of the double-stranded protein. This is the signal you see here. And as you can see here, actually, the enzyme works much better if you put the other two cofactors. So you have the RNA polymerase, but then you have NSP7, NSP8, 7-8, that are the cofactors that are really stabilizing the protein and making it work better. And this is the molecular, the structure of the RNA polymerase recently published. A second assay we established is an assay for the helicase. In this case, helicase is a protein that opens up the RNA, is a 5' to 3' helicase. So basically, what it does is, this is the substrate that it can be recognized. The helicase binds to an extremity here, and in the presence of ATP, opens up the double-strand RNA. The assay is as such that, as I was mentioning before, you have a fluorochrom, but also a quencher. So the substrate basically is quenched, so you do not have a signal. But when you have the helicase, you have the separation of these two. And so the fluorochrom is now far away from the quencher and you can measure the signal. And to stabilize this product, we also add an extra an extra sequence that is similar exactly like this, but it is much in much more abundance. So once this RNA signal is sequence is open up, does not anymore go back to quenching the signal. And this is just a graph showing you how it's easy to make this assay. And so in this way, what we are doing now, we are using a number of libraries to screen basically a few thousand candidates against the polymerase and helicase, hoping to find some better results at the protease. That was the first and the main object of the study done until now in the entire world. Of course, then we will need, of course, probably a lead optimization, and so to do the ADMET study to go back to the clinical studies. And of course, this is not COVID-19. This is not very updated, but is recently updated. We don't have only COVID-19. We just remind you, we have Zika, we have Ebola, we have, of course, influenza, we have still MERS. So there are a number of, let's say, there are a number of pathogens that have been listed by WHO for the need of drug development. Among the worst are Ebola and Marburg virus against the coronavirus, SARS and MERS, but also Nipah virus, Lassa virus, Zika virus, but not to mention dengue or West Nile, chikungunya virus, and of course for some, more far away, it's not a big deal now for Herpes virus. So anyway, this is the long way of approval of the drug. But then the last slide I want to show you basically is this. How can we advance in this? Well, we need really, in my opinion, a coordination between funding agents, a consortia with big partnership, and then a consortium made of academia, government, private sector, NGO, and cross. And so, all I'm going to finish this my final slide. I hope I show you that we need drug against SARS-CoV-2. We can have different approaches, both a target-based or a phenotypic approach. Of course, as I was mentioning before, the drug repurpose has not been very effective until now, but doesn't mean that it doesn't work for every virus or very disease. I mentioned briefly, I didn't really go into it, but of course, the issue of pan-coronavirus efficacy, and this, of course, brings you an issue that I didn't really mention, but is the similarity among the targets. There are some targets are more similar than others among the coronaviruses. Of course, then we have an issue of more drugs or multiple targets, as you know, for both HIV and HCV. We have, let's say, in one case, made chronic a lethal disease. For HCV, we are going versus let's say, a cure of the patient, but always with multiple targets, with multiple drugs aimed at multiple targets, because there is an issue of viral resistance that, of course, this is not studied yet, but of course it will. So drugs with broad spectrum versus different emerging pathogens. Another issue that is important to keep in mind, I mentioned your remdesivir that come from Ebola studies, Ebola virus studies. So meaning that a broad spectrum acting agent is of course very welcome. We need a partnership, and of course we need a global access to treatment for the preparation for next pandemic. With all of this, I really thank you very much. I end my presentation here, and I hope I can answer to the question you might have. Thank you very much.
Thank you very much, sir, for your kind informative lecture on SARS-2 COVID virus replication of new drugs. You had explained very nicely about the protein drug binding, drug interaction, and the important factor of protein receptor binding of new drugs that would be important for COVID virus. So thank you very much, sir. Now I would like to request Dr. Anil Malagutti, sir, Dean Extension Affair, to propose the vote of thanks. Anil, sir.
Yeah, thank you, sir. Before that, I would like to ask if the participants have any queries, so they can unmute and ask themselves. Any participants? So if any queries are there, so sir will answer. So we have not seen any questions in the chat box. Sir, it was very well explained. Indeed, it's great honor for me to propose the vote of thanks. So I, Dr. Anil Malagutti, Dean External Affairs from Sakalchand Patel University. So here we come to an end of wonderful session. On behalf of Sakalchand Patel University, I would like to extend my vote of thanks. Firstly, I take an opportunity to thank Sri Prakash Patel, sir, President of SPU, for providing us such an excellent infrastructure and opportunity. Providing us with an excellent infrastructure.
There we go. Now I hear you. I don't hear you though. You have to turn on your microphone.
Hello.
Hello.
That's
Perfect. Yep. Sorry.
Yeah.
Sorry. That's okay.
Yeah. So with this, we come to an end of a wonderful session. On behalf of Sakalchand Patel University, I propose a vote of thanks. So I would like to thank firstly our President, sir, Sri Prakash Patel, sir, for providing us an excellent infrastructure and motivating us to conduct the international multidisciplinary webinars. And he is also supporting us to organize the more activities for the academic purpose. I would like to thank Dr. J.R. Patel, sir, Provost of SPU, for his keen interest and boosting energy for the entire show and encouraging all faculty members and including me, like supporting us for organizing this webinar. I thank you, sir, for your constant support. So I would my greatest thanks to today's honorable speaker, Enzo Tramontano, sir, for wonderful scientific deliberation and sparing his time from very busy schedule. I am in touch with him since I think last 2 to 3 months, and because of the pandemic, we could not arrange sir in between. But I am very grateful that ultimately you agreed upon this, and we are very thankful to you for sparing your time, sir. My special thanks to Dr. DJ Shah, sir, Director of Technical Courses. So always before planning for any webinars, he guides me and he supports to organize the webinars. Thank you very much, sir. And I thank Dr. J.K. Patel, Dean of Nutan Pharmacy College, for his meticulous implementation and planning of this webinar and support which he has provided. I would like to thank Hirak Joshi, sir, Professor, Department of Pharmacy, and his team for organizing this webinar. I would like to thank IT Department of our SPU and all the participants. We received a large number of participation from across the India and from outside the country for this webinar. And this webinar was an excellent deliberation of your lecture, sir. Thank you very much, sir. Thank you all. Yeah. Hello, Enzo, sir.
Yes, I'm here. I'm just.
Yes,
I
sir.
thank you for. I thank you for giving me the
What I
opportunity.
request, yes, sir. Enzo, sir, what I request, I will end the meeting and restart it so that to our provost sir and other head of the institutes, deans are will be joining again.
Okay.
Yes, sir. DJ sir. सर एंड करके ज्वाइन कर दूं वापस